¿Se escucha? ¿Se escucha? Sí. Buenas tardes a todos. Bienvenidos a otra ocasión de nuestro seminario de Frontiers in Genomics. Esta vez tenemos al doctor David Gilbert. El doctor Gilbert estudió bioquímica y biología celular en la Universidad de California, San Diego, y hizo un doctorado en genética en Stanford. El trabajo del doctor Gilbert se enfoca en los mecanismos de regulación de la replicación del ADN durante el ciclo celular y la relación de esta replicación con la organización estructural y funcional de los cromosomas. Actualmente él está trabajando desde 2021 él está trabajando en San Diego Biomed como profesor, y él ha sido un investigador principal en el NIH Consortium y es un miembro de NIH 40 Nucleosome Consortium y el Southeast Stem Cell Consortium. Y pues, sin más dilación, empezamos con su presentación.
Ready? Listo. Okay. So thank you very much for this invitation to this beautiful city. It's my first time to this middle of Mexico, not the coast, any kind of coast. So it's very different. It's been enjoyable. So I'm going to talk today about studies linking the three-dimensional organization of, whoops, I'm pushing the wrong button, how replication is organized in mammalian cells in 3 dimensions with how it is organized along the length of the chromosome. So here, what I'd like to say, I'm trying to center this thing here, there we go. So replication is a really wonderful opportunity to combine or integrate data sets for genomics with data sets for imaging, which is a major task right now in 4D nucleome studies and genome architecture studies, is being able to actually see in a cell what we measure by genomics. And so that there's a big effort for that. And what you can see here is that in this image below, it shows that if we label cells with nucleotide analogs early in S phase and then chase them, wash out the nucleotide analog, chase them into late S phase and label with a second different nucleotide analog, and then differentially label them, you can see that replication takes place throughout the interior of the nucleus in early S phase and at the periphery of the nucleus and around the nucleoli later in S phase. You can also see that replication in very brief pulses takes place in these punct-type foci that we call appropriately replication foci, just a descriptive term. But we believe that those may be individual domains that are independently regulated. And up here, you're looking at genomics data that I'm going to explain in detail later, but you're looking at early in green and late in red regions of a chromosome along the length of the chromosome. So you can see there are early replicating, early initiating, middle initiating, and late initiating regions throughout the genome. And that there's also discontinuity. So there may be small regions within here that represent these foci, but that hasn't actually been proven. So you can imagine lots of opportunities then to link what you see and what you measure by genomics. Okay, so I'm going to take you back in time to the very beginning, J. Herbert Taylor's experiments. J. Herbert Taylor synthesized tritiated thymidine in the 1950s, and then he basically laid out all of the basic concepts of DNA replication that we know today, including semi-conservative replication and anti-parallel replication, and long before Messelson and Stahl, if that's who you think of. And so what he's done here in this experiment is to briefly label cells with tritiated thymidine, which then goes into DNA in the place of thymidine, and then chase the cells for different lengths of time into mitosis. So cells that were chased longer into mitosis were in early S phase at the time of the label. Cells that were chased for shorter periods of time were in late S phase at the time of the label. So what you see here is a late S phase pattern. And what he saw here was 2 different things. One is there were very large regions of the chromosomes by kilobases, as we call it now, but by size, that were labeled within these very brief periods of time during S phase. But secondly, and most importantly for our talk, he found that the patterns of labeling on the chromosomes were recurrent and specific for particular times during S phase. So there were early, middle, and late labeling patterns along the length of the chromosomes. And so what you're looking at over here on the right is my first experiment as a graduate student, where I had learned about antibodies against BRDU that were being used for other reasons, being used actually to study DNA repair in another institute. And I got a hold of these antibodies, and I reproduced the exact experiment shown here. And you can see that I could also reproduce these patterns. And if you're looking for an easy one, you see the Y chromosome, it's almost completely replicated within a 10-minute labeling period during S phase. Okay? So the entire Y chromosome is late replicating. So I was able to do literally in a few hours what took J. Herbert Taylor months, because autoradiography and development of tritiated thymidine took that long. However, I want you to also see that this is about 25 years, and this is about all the progress we made. Okay, so from the questions that were defined by J. Herbert Taylor to what we know almost now, but certainly back in 1983, what regulates this program? He proposed that it must be genetic because it's recurrent. What regulates the initiation of replication? What regulates when things replicate? And what is this biological significance of this program? Okay, so those have been sort of the questions that have driven my career. So I went on during graduate school to develop a method to measure molecularly replication timing. And I wanted a method that I could apply to any cell type, independent of synchrony methods or other individual idiosyncrasies of particular cell types. So what I came up with was to pulse label cells with bromodeoxyuridine, just like I used in the last slide with antibodies, but then to stain the cells for DNA content and take them to the fluorescence activated cell sorter, and then ask the cell sorter, and when you stain for DNA content, of course, you get a large peak of cells in G1, and then cells with increasing DNA content that are in S phase, and then another peak for cells that are in G2 and M. I could then ask the sorter to sort out any number of times in S phase, but our most robust method, our simplest method, is we take the first half and the second half of S phase, isolate the DNA, immunoprecipitate the BRDU substituted DNA, and then in today's day and age, we sequenced it. Of course, when I was a graduate student, we didn't even have PCR, so I was hybridizing. Later, we had PCR towards the end of my graduate school period. And then in the beginning of the 21st century, we started to have microarray and then sequencing, okay? But in all of the cases where we do genomics, microarray and sequencing, we display the data as a log-to ratio of enrichment of any given bin along the length of a chromosome in early to late, okay? So higher on the y-axis is going to be earlier replicating, lower on the y-axis is late replicating. You're going to see a lot of plots that look like this, okay? Okay, so here's the raw data that we get coming off a sequencing machine in 5 KB bins. And you can see right away that there are early replicating domains, late replicating domains, and middle replicating domains. And you can see that these domains are punctured. They're not really, I should call them constant timing regions. I should not call them domains at this time, but they're regions where large swaths of the chromosome replicate at relatively constant times. And then you can see that these are punctuated by what we call temporal transition regions, where replication is proceeding linearly with time along the chromosome. And for the most part, with some exceptions, these regions are moving consistently with a single replication fork, moving very long distances through the genome at 2 kilobases per minute, which is the known rate of elongation, okay? So this is kind of a dangerous situation because you have this fork out there that's chugging along for hours and hours, replicating hundreds of kilobases. Okay, so then just to summarize, we can, since adjacent regions replicate at similar times, we can then smooth these data and segment them and then ask questions about how many domains there are and how big they are, et cetera, where they are, where their boundaries are. And to summarize a lot of work over the years, we showed that this program is incredibly robust. We could knock down all sorts of chromatin regulators, treat with all sorts of drugs that are inhibitors of chromatin enzymes, and this program is not affected at all. However, as soon as we would differentiate embryonic stem cells, either mouse or human embryonic stem cells, we'd get a massive reorganization of the temporal order of replication. And if we look over, about 30 different cell types in mouse and 30 different cell types in human, we find that about half the genome changes during differentiation and the other half doesn't. So one half is constitutive, one half is developmental regulated. And we found early on that these changes occur in units of 400 to 800 kb. So by that, what I mean is, whoops, my little cursor got stuck somehow. Come on. Oh, okay, that's weird. Sorry. I'm trying to use a pointer that people that are in Zoom will be able to see, but yet still be getting away from this podium so that you can see the slide. Anyhow, so if we just take like a large domain, like a large region, I keep saying domain, that's a mistake. A large region like this, the 5 megabases, that replicates relatively constantly in embryonic stem cells. If we look at other cell types, parts of this region will be replicating late. And in other cell types, other parts would be replicating late. And then they would switch back to early, et cetera. And what we found was that when we see these switches, early, late, in different cell types, they're always occurring at the same boundaries. And they're always in this smaller size range of 400 to 800 kb. So that gave us the first indication that we had a unit of replication regulation that we called replication domains. Okay, and as probably all of you know from textbooks, these changes are coordinated with changes in transcription, chromatin, and 3D organization. We showed that back in 2010. And by this I mean late replicating genes are difficult to reprogram. By that, what I mean is when you make induced pluripotent stem cells from fibroblasts, you get lots of colonies. Some of these colonies are bona fide pluripotent cells, but some of these haven't made it all the way back. So they look like pluripotent cells, but molecularly, they have not reactivated a lot of genes that are pluripotent specific genes, okay? And what it was thought, everybody would just throw those colonies away because it was thought that those were artifacts of the reprogramming process. But we decided to take a look because another hypothesis is that cells run into a brick wall when they're reprogramming. And they're all actually lining up at some place they can't get past. So to test that hypothesis, we did RepliSeq, this replication timing method, on multiple clones, multiple partially induced pluripotent stem cells. So multiple individual clones, separate isolates that were not able to make it all the way back. And we found those had exactly the same replication timing program. And it differed from fully reprogrammed cells in very specific regions. And those regions were regions that are early replicating only in pluripotent cells. And they contain the genes that are failed to reprogram in partially induced pluripotent stem cells, okay? So while this is all correlation, everything you guys are gonna do in genomics is a correlation. Always remember that. Mother Nature will fool you with all kinds of correlations until you do causality experiments. You haven't proven anything. However, this is pretty good, some of our best correlative evidence that late replication represents an epigenetic barrier to reprogramming. So if something's late replicating, it's difficult to flip it back and change cell fate, okay? Okay, moving on now. More recently, we've developed higher resolution and single cell methods to measure replication timing. And so with what's shown here is our high resolution RepliSeq. And very simply what we do is we basically prepare the cells the same way as for our what we call E over L or log2 ratio method. But instead we sort 16 fractions of S phase. And instead of making a log ratio, we sequence each of these fractions individually and then make a heat map of the density of sequences in each of these temporal windows from S1 down to S16. Okay? And so you can see that this profile resembles very much the lower resolution E over L, but it has a lot more detail. And one of the things that we could decipher from this is how deterministic the replication timing program is, because you can see that the vast majority of reads are within very specific temporal windows. However, there's variability in this. So you can see that this region here can replicate at any time throughout the last quarter of S phase practically, okay? And you can see variabilities in the thickness along, so pardon this large laser pointer. I worked for like 10 minutes trying to make it smaller and I gave up. But anyways, you get the picture. And so we can measure that as what we call T-width, or the time, that is the S phase fractions, over which any given bin is distributed, okay? Now, we can also do single cell repli-seq, because remember, this is still a population-based method, but we can still tell heterogeneity in the population because we have such sharp temporal resolution. Real single cell data is shown down here. The problem with this data is it's much lower resolution, but it is direct cell-to-cell variation. And all really single cell repli-seq is whole genome sequencing, because early replicating sequences are going to be more represented than late replicating sequences. Okay, so when something replicates, it's twice the copy number as when it doesn't, before it replicates. So what we can do is do a whole genome sequencing and then align cells over here on the left in gray by their DNA content. So that lowest DNA content to the highest DNA content. And then we simply go across here digitally, and in red, say the bin has replicated, or gray, the bin has not replicated. And what you can see is that early replicating sequences, here's the E/L repli-seq at the bottom. Early replicating sequences are already replicated at very low DNA contents, and the late replicating sequences don't replicate until much higher DNA contents. And we can also measure heterogeneity in these, in single cells by looking at the a deviation of replication timing amongst many cells and many different bins from its average. So on the y-axis, on the x-axis here is the 0 is the normalized average replication timing from the log2 ratio. And then we look at each bin and look at how much it varies from the 0. And you can see that it's still quite deterministic, even at this lower resolution. Here's what deterministic would look like, and here's what random would look like, okay? So now keep in mind, as I told the students yesterday, that single cell data, I don't care what kind of data it is, unless it's in haploid yeast or haploid cells, it's an average of 2, okay? So single cell data is not single event data. But you can make it single event data if you use haploid cells. There is one human cell line that's haploid that we're allowed to work with, because the other one is made by parthenogenetic activation of human oocytes, which is not allowed in US for NIH-funded labs. So there's only one cell line left, okay? So that's pretty restricted. And with mouse, I told the students yesterday, we crossbreed 2 mouse subspecies that are separated by 500,000 years. They have a SNP every 150 base pairs. And so therefore, we have a SNP almost every read, and we can then separate the genome. So we can get true single molecule data from these, okay? But when you see single cell data, it is 99.999% of the time not single molecule data, okay? All right, moving on now. Okay, how is this program regulated? So going back to the beginning days of my lab, we didn't have CRISPR, right? But we didn't even have sequenced genomes back in the 90s. So we couldn't really look at very easily at replication molecularly. But we could look at it cytogenetically for the reasons I told you in the first slide. So what we did in this experiment is to label cells, just like I showed you in the first slide, with one nucleotide analog early in S, chase, label with the second one late in S, and then chase them further into mitosis, where you can see the bands on chromosomes that J. Herbert Taylor was looking at. And then we could ask 2 different kinds of questions. One is, how long does it take for these labeled early and late replicating regions of chromosomes to go from bands on chromosomes to their positions in interphase, where they will then reside for the rest of the cell cycle? And so what we did was simply follow them, and what we found was that early in G1, they were scrambled randomly throughout the newly formed nucleus. So this would be after the nuclear membrane is formed. And if we transfect in fluorescent nucleotides, which we can't just, they won't go across the membrane, but we can transfect them in, we can look in living cells. And what we could see is that at these early stages of G1 phase, these foci were moving around, highly, highly mobile. And then at a time about 2 hours after nuclear envelope formation, they would then become anchored at places in the nucleus where they would then reside for the rest of interphase. So the next thing we could do with this in this experiment is to take nuclei from cells at different stages during G1 phase and introduce them into a cell-free replication initiation system that I developed as a postdoc. So this system could jumpstart cells or nuclei, purified nuclei, in vitro, could jumpstart S phase. And by doing that, what we could do is ask questions about the preparedness of the nucleus for carrying out the proper replication program, right? And what we found was that with nuclei at this stage of G1 phase, replication proceeded randomly in a random temporal order in our cell-free system. And then shortly after nuclear envelope formation, when this 3D structure had finished forming, replication proceeded in the proper temporal order. Okay? So we called this time in the cell cycle the timing decision point, the time at which replication timing is established, and it's coincident with the reestablishment of the 3D organization of the domains in the nucleus. So this was the tightest correlation between these two events. Now later, many years later, another graduate student of mine wanted to know, when is this program lost? And our hypothesis was that it would be lost at mitosis, because that's when all of this 3D organization is taken apart. But in fact, he found a big surprise to all of us that nuclei in G2 phase, when jump started to initiate replication either in vitro, or we then set up a system to get cells to trick cells into thinking they'd gone around the cell cycle and to reinitiate in G2 phase in living cells. Either case, we got a completely random replication timing program. In fact, in vivo, we could get 2N on the facts, 2N first S phase, 4N second S phase, 8N. We could sort in the same group of cells, the first S phase, perfect temporal order, second S phase, random. Okay? So what we're looking for, this tells us 12 years ago, we're looking for some kind of determinant that's assembled at this time in early G1 and is somehow lost during S phase. Probably at the replication fork, something is lost that dictates timing. And we're still looking for something with those properties. Okay, so we proposed this very naive model back then, that whatever it is that regulates replication timing, maybe it's disassembled during mitosis, and then as domains or as regions of chromosomes can become anchored, once they become anchored and they're immobile, you can then assemble microenvironments in the nucleus of different molecular composition, right? And what we called those, what we said was it could seed the assembly of subnuclear microenvironments, increasing local concentrations of factors that modulate pre-replication complex activity, okay? Today, you probably know of this famous thing called phase separation. This is exactly that. Some people call phase separation seeded nucleation. But basically, this is exactly what we're talking about. Once you're anchored, you can start to attract molecules, and self interactions would then allow for a stable structure to be formed. And the paradigm back then wasn't a novel paradigm. The paradigm was the nucleolus, which we knew a lot about. It was held together without membranes by self-associations of proteins that are zipping in and out in an incredible rate, okay? So they're not separated by membranes, they're not solid structures, they're not static, but they are nonetheless microenvironments. And then we proposed that these microenvironments could then set thresholds on replication so that low thresholds would fire first and high thresholds would fire later, based on whatever the composition of these microenvironments was. And I'm going to show you in a little bit later in the talk the modern version of this model, okay? But we still hold to this model. Okay, this is an old slide, but what I wanted to show you is that when we look at replication origins, that is, the sites where replication initiates, what we find is that after the timing decision point, replication is initiating in the proper temporal order when nuclei are introduced into our cell-free system. But we could not detect any specificity in the initiation sites whatsoever by the crude methods that we had back in the 90s. What we found was that if we keep isolating nuclei at later and later time points, we could find a time point after the timing decision point, before the restriction point when cells commit to going into S phase, at which replication origins are now specified to the degree that we could measure them back then. So that means two things, that what determines replication timing is independent of and upstream of the origin of what determines where you initiate replication. And we can isolate nuclei that have passed one point but not the other. So clearly determining timing is not sufficient for determining origin specification. Okay? So what we still adhere to is that the timing is mejor, as you would say, to in origin, that the timing is set first, that's the more important of it. And then where you initiate replication clearly doesn't matter because you can initiate at random here. And we were even able to trick cells into doing this in vivo, in culture, in live cells, and initiating replication without any origin specificity. Okay, so let's talk about origins. They were mapped back when we did these experiments. It was there was this huge controversy going on, still going on, as to whether origins are site-specific or completely random. And obviously, nothing is one or the other. Everything is some sort of compromise. But people would sort of throw out some data or the other. And then some people would use my findings as a way to reconcile them, or maybe it's chromatin or something. But in fact, the way to reconcile them is that different methods were picking up different aspects of replication origins. And that replication can initiate at almost any site in the genome, but that it's stochastic. So there's a probability of it initiating at different sites. And some methods are able to only see the tip of the iceberg, and all they see is only the most highly probable to fire. And so then people think those are site-specific. But people who have methods that are able to detect low efficiency origins, they see it happening all over the place. Okay? Now, so the hypothesis then is that it's highly stochastic. The only way to prove a stochastic process is to have single events, single molecule events. So when BioNano came out with its platform for mapping long DNA molecules which is basically done by taking restriction enzymes that are deficient in their ability to cut DNA, fusing them to enzymes that can then covalently link a fluorescent molecule to DNA. And in that way, you can put a fluorescent molecule wherever their restriction site is. And then you run them through nanochannels at an extremely high throughput and take photographs of them, and the computer tells you the restriction map. So when this technology was developed, we said, aha, well, all we need to do is to transfect fluorescent nucleotides into cells that are synchronized right at the beginning of S phase, which we knew how to do really well, so that we're looking at the first nucleotides to be synthesized at the beginning of S phase. Those are by definition the origins, and then just see where they are. And that's what we did, and it worked amazingly well. And we published this paper about a year ago, 27 million DNA fibers. The previous study that had been done in 2012, the biggest study ever done, was done by individually stretching fibers. It had 274 fibers. Okay, so we went from 274 to 27 million with this technology. And what you can see here is the display. Each gray line is a fiber. And the little ticks you can sort of kind of see on the slide inside are the places where replication initiated. And fibers that were not labeled are, you know, all the way off the floor in this slide. And what you can see is that replication doesn't initiate at random. It likes to initiate in certain zones, as we call them, initiation zones. But within those zones, there are tick marks, you know, pretty much in many, many different places, okay? But you can also see that almost every part of the genome along the length of this segment has some DNA fiber that has an initiation site, okay? So since we are looking at all the fibers, both the fibers that are labeled and the fibers that are unlabeled, which is not able to be done by other methods, we were able to get actual efficiencies. So what's shown here is, remember, we caught the very, very earliest origins to fire. So we plotted replication timing of all of these different initiation zones that we found versus their efficiency. That is literally the percentage of times that they were fired, right? Percentage of time that they were used as an initiation. And you can see that the very, very earliest regions fire between 5 and 20, and this 20% is an outlier. You can see it's way up here. So fire mostly between 5 and 15% of the time, okay? But it's not random. They all have a different probability of firing. You can also see over here, if we, since our resolution is about 14 kb, we can predict from these approximately 40 kb tracks of labeling that we see, how many different initiations could possibly be in there based on our data, okay? So if there's an initiation site here, here, and here, you know, and our resolution is only 14 kb, you can mathematically determine the minimal number that must be in there. And the median of that is 4. So there's at least 4 sites per 40 kb, and that number may increase as we get better and better resolution, okay? Okay, so replication timing is highly deterministic. Origin selection is highly stochastic. So, but it's not random. So what determines where replication initiates? And we know very little about that, but we know a couple of things, and these are actually some old data from my lab, and I'm going to kind of go backwards through time with it just to be logical. But what we know is that replication initiation must de facto occur at the sites where there is mini chromosome maintenance helicase. So the helicase, eukaryotic helicase, all eukaryotic helicases are called MCMs, okay? It's a heterohexameric ring. It's loaded onto double-stranded DNA in telophase and continues to be loaded throughout G1 phase. So we know wherever replication initiates, there has to have been an MCM there at that time, okay? And what we also know from a study done by a former graduate student of mine, lost my pointer again. Maybe Amazon has a better pointer that I can use for the next talk. I'm not sure if I'm going to lose it entirely. Didn't this happen yesterday? Like it just stopped working on its own? Anyhow, if I point at it, I'll lose the people on Zoom. So let's go to this sort of difficult to maneuver with here, but I'll use this this cursor. Okay, a former graduate student of mine showed using biophysical techniques, that is photobleaching, where you label the MCM fluorescently, and then you photobleach the nucleus so that you put, with the laser, you eliminate all fluorescence within a particular region. And then you ask how long that fluorescence takes to recover, and that recovery has to come from fresh molecules, okay? So how fast is the exchange? If you do that with any chromatin protein, it's going to be incredibly fast, okay? With MCM, we discovered MCM was the most stably bound protein to DNA of any protein known to man before or after us, okay? She could write her name in the nucleus, come back the next day, and if we didn't let the cells enter into S phase, everything would be still there, clear as a bell. Zero exchange, okay? So we know it's locked down, okay? But we also knew that the origin decision point, I'm sorry, I'm just instinctively using this pointer. The origin decision point was sensitive to transcription inhibitors, okay? So we proposed this model that as the MCM is loading during G1, When transcription comes on, it's sliding the MCMs out of the transcripts, okay? And we know that initiation is depleted in genes and enriched in intergenic regions, okay? And then later, it's been actually, come on, cursor, there you go. And it's actually been proven in yeast using genetic techniques that this is the case. And then additional data in Drosophila and mammals has come out that this model is indeed correct. Okay. Next model, fast forward to today. We have a paper hopefully in press with Jennifer Cremens, where we believe we've provided evidence that chromatin extrusion is also able to push MCM into the regions between topologically associated domains. So now let me back up, because I know there might be some people in the audience that are not familiar with high C and topologically associating domains. So essentially, high C is a method to measure the three-dimensional organization of chromosomes. And what you do is you take a population of cells, fix them with formaldehyde to covalently link everything to everything else, freeze it. Then you cut with a restriction enzyme and religate. So any DNA that's in close proximity, some people call this proximity ligation. Any DNA that's in close proximity will become covalently linked by ligation, okay? You then just chop up the DNA and sequence it, and you look for reads that have what are called split reads, that have one sequence from one part of the genome and another sequence from another part of the genome. And from those points that you know are in close 3D proximity, you can rebuild a model of the 3D structure of the chromosome. Again, this is the most common way of displaying that type of data. And the way it's displayed is that you have all of the restriction sites of a chromosome along the x-axis and all of the restriction sites along the same chromosome along the y-axis. And then each dot on the matrix is one read that you got from your sequencing. So you can see that there's a ton of reads of sequences that are very close together, because they are close together. They're close together along the linear length of the chromosome. But you can see that as you get further away, the more distant you are away, the further you are from the diagonal, you see weaker and weaker interactions, but they're not uniform. So what you can see, first of all, is that there are these regions of self-interaction. And these are the topologically associating domains. Along the length of the chromosome, there are regions that prefer to interact with themselves. And you can also see longer range relationships of regions that are more, less, tend to associate. Okay. Now, some of these self-associating regions, or TADs, we're going to call them TADs from now on, have these things called corner dots at their corners. And what those are believed to be is regions of very strong interactions held together by cohesin. Okay? So you guys may know cohesin from textbooks as holding sister chromatids together, but it also has a function of extruding chromatin. So it's a ring-shaped structure, and it will take chromatin and pull it through itself, or it'll slide along while the chromatin is pulling through. And it will slide until it hits a CTCF site. And it actually has to hit two CTCF sites in the same orientation before it will stop. And when it stops, it pauses, and that creates these dots, okay? Because now you have high frequency interactions. So a high percentage of the reads have linked the two sequences that are linked by cohesin, okay? So these are called dot domains. What we can then do is take all of these TADs, and we can catalog them, whether they have dots or not dots, and there are other ways we can catalog them. But then we can take all the TADs of a particular type, make them all the same size computationally, and then pile them all up, okay? And when we do that, and we align those aggregated or piled up TADs that are dot domain TADs to our 16 fraction repli-seq that's also piled up, what aligns with the boundaries of these TADs is the very earliest initiating initiation zones, the very earliest regions to initiate. How do we know this has anything to do with extrusion? Well, we knock out Rad21, and when we do that, we lose this alignment, we lose the dots, and we lose the alignment, okay? So Rad21 we already knew from previous work was required for the dots. Now we show it's required to keep those origin, or to focus those initiation sites close to the origin. This part's in bioRxiv, by the way. So you can go find it if you want. Okay, so, and we also went on to perturb other components of the extrusion pathway and show that it's actually extrusion activity. Okay, so we think replication timing is chosen first in the cell cycle. We think it's chosen at some level of assembly of subnuclear domains, these microenvironments. And then we think the choice of where you initiate within that microenvironment is stochastic. It's not super important, okay? But we'll never be able to test everything I've shown you, is some correlations, and then everything is indirect. We're deleting Rad21, we're inhibiting transcription, and we're looking at initiation. Where are those helicases? We're never going to be able to address any of these questions directly until we can map the positions of the human replicative helicase. Can you believe it's 2022 and we don't know how to find it? So the hypothesis that we've been working with for a long time is that it's difficult to map because it's binding all over the place. So if you scrape 1 million cells down and you do a chip, you're just going to get noise. Okay? That's the hypothesis. But in order to test this hypothesis, you need a way to map protein binding in single cells, and that hasn't been easy. In fact, it's been impossible. Every few months you might see a new paper that says it's doing it. Then you look at the data and, well, it's kind of disappointing. In some cases, histones have been mapped in single cells at some reasonable amount. The data look reasonably okay, although it's always very sparse. So Dan Bartlett, he joined my lab, and his job, what he wanted to do was to map MCM, but he knew he had to come up with a single cell mapping method. And so he spent his entire thesis doing that, okay? So, but what he came up with actually really works. And it was quite simple. After working with every method that was out there, and then, being a master of the literature, as all you graduate students have to be, what he realized was that he could combine cut and tag with what's called linear amplification. Now, I'm not going to go through this in detail in the interest of time, because we're now 40 minutes into the talk. And you can read the paper, it's published, okay? But what I'm going to say is the trick was very simple. So you use, in cut and tag, you use TN5 transposase to insert a barcode. You fuse the transposase to protein A, and then you use your primary antibody to find your protein, and then you use the protein A fusion. The protein A binds to antibodies, and then the TN5 inserts a barcode wherever it's hanging out. All Dan did was put a bacteriophage T7 promoter in front of the barcode. That's it. And by doing that, he was able to do linear amplification by adding T7 polymerase, avoiding all the artifacts of PCR, which if anyone's done cut and run, you know what those are, and getting much larger percentage of the genome to be read out. He only needed one barcode to be inserted, and then he would get transcription coming off of that barcode. Reading that barcode. So we get literally 10 times more unique reads per cell than any of the methods published before or after. And that has allowed us to get into single cells, because previously single cell data, as I said, was sparse, meaning you only get a bit of the genome and you're missing other big parts of the genome. So in this case, single cells look almost like bulk in their readout. And so Dan's done, and actually he's pretty popular. So he's getting a lot of job offers just for doing that. And so Rani Fuwanti has joined me in San Diego, and she's picking up where Dan left off. So I suspect I'm going to give her about a year before we get the first real images of MCM. Okay, so now I'm going to move on to 2 more published stories. The first one addresses this question. Back to this question, what regulates replication timing? Okay. And so this brings up a graduate student, Zhao Sima, who started in my lab just about the time CRISPR started. And she said, I want to identify the cis-reacting elements of replication timing. Well, at that time, I had reviewers and other people disagreeing as to whether there was even a genetic component to replication timing. Maybe it's all epigenetic. It's all transcription. It's all just a reflection of other activities of the genome. So that was risky in itself. But she's trying to identify something that had been tried to identify for over 50 years, right? And some really good labs like Mark Brudine's really tried hard to find the cis-acting elements regulating replication. But, she correctly said to me, but I have CRISPR. So go for it. So she went and she made, somebody counted, it was over 100 deletions. But at a certain point when she had made dozens and dozens and dozens of deletions, the data just made no sense whatsoever. So we weren't, we were kind of at a stuck point because all we had was partial effects, reproducible partial effects, the worst nightmare of any graduate student, right? So what we reasoned was that, well, we know replication is related to the 3D organization of chromosomes. So let's get the highest possible resolution 3D structure of this domain. And then let's see what it looks like. And what we found was that there were 3 points in this domain. This is a domain that's replicated early in embryonic stem cells and late in all other cell types. And we looked in detail at this domain. And we found that there were these 3 points that were interacting in space. Those points did not contain CTCF. And Zhao herself had shown earlier that CTCF knockouts have no effect on replication timing, which was kind of irritating to all the, budding genome architecture people who thought CTCF was everything. But it clearly it was not required for the replication timing program. So therefore, clearly not required for compartments in our interpretation. They had broad peaks of H3k27 though, and they were interacting. So she deleted all 3 of them, and this is the result that we got. Okay, complete shift from early to late. So this was really if I had to pick one image for my entire career, this would be it. And this is really the biggest discovery because we have now discovered the enhancers of replication. And we can discuss that later if you like, but we do not think they're the origins. We think they're the enhancers. And in addition, this was satisfactory because we could explain why it took 60 years to find them. Because what Zhao did was went back and deleted them 1 at a time or 2 at a time and found that if you delete 1 of them, there's no effect on replication timing, or C has a small effect, but she never hit on C in all of the deletions that she had done. And if she deletes 2 of them, it's still the earliest replicating region in the genome. And an interesting anecdote is that she had a 680 kb deletion that took out all of this DNA from the yellow all the way over to like here, and it looked exactly like this. So it was very puzzling that we could take, 3/4 of the domain out and still replicated, in the middle of S phase. So anyway, these deletions explain why it took 60 years, because there's redundancy. And it was hard enough to make 1 deletion before CRISPR, but to then find there was no effect, would you actually try to delete more? It just never happened. And it also explained all of her confusing results from the first several years of her PhD. Okay, so Jesse Turner's a newer graduate student in the lab, and he's basically doing, just bashing these up, trying to make the smallest deletions possible that contain the activity. So I'm showing 1 example here. This happens to be one of the biggest deletions of Zhao's that Zhao made, 40 kb, that had, I think this is the C element. And if you look at tracks of transcription factors, enhancers, et cetera, you can see that the business end looks like it's over here. All the transcription factors and enhancers are here. And so, in fact, he did a series of deletions. And in fact, this was the business end of the region. But he also showed that if he deleted these 2 OCT4 SOX2 nanog sites, OCT4 SOX2 nanog are transcription factors that are the core factors at at the core of transcriptional regulatory networks in pluripotent cells. And if he knocked out regions that bound all three of them that we call OSN sites, he could recapitulate the phenotype of the entire 40 kb deletion. If he deleted the transcription start site, he got no effect on replication timing. And I haven't said, I mean, it's published. This isn't published, but the Zhao's work is published. When you knock out all three ERCEs, you get this complete shift from early to late. You also get a compartment A to B, for those who are familiar with subnuclear compartments. You get a shift of the whole locus from the interior of the nucleus to the periphery of the nucleus, and you lose all transcription throughout the region, confined to the region, though, not affecting anything outside the TAD. So what we've been trying to uncouple these, and this deletion here shows that we can uncouple transcription and replication timing if we hit the right spots within these ERCEs. The other thing he's doing is transferring them ectopically to a very large late replicating region shown here. And this just shows you if you move this A element, which is actually the smallest one Zhao found was 3 kb. If you move that 3 kb over here, you get an early middle replication pattern, which is exactly what you get if you delete everybody else and leave A alone at the endogenous site. So this tells us that we can transfer the activity, and now we can do sufficiency assays, which I think are going to be a lot more powerful for us. But it also shows you that DNA sequences carry the information because these sequencers are taken out, cloned in E. coli, purified, and then put back in to another location. Whereas our deletions are done in the chromosome, and we could be deleting an epigenetic mark. It could have been epigenetics still. Okay, to summarize this story, what we think is happening is we have these OCT4-SOX2 nanog sites that are and we think that somehow they are related to these high levels of histone acetylation and the ability of these regions to interact. We don't know how those things happen, but there are anecdotes in the literature of how OCT4-SOX2 and nanog could recruit histone acetyltransferases or could interact with each other. Those data aren't very good, but this is our working model for now. They do come together. They are OCT4-SOX2 nanog, and they are bound with high levels of histone acetylation. Well, when you bring all three of them together, you're now making a hub that's super concentrated with histone acetylation, okay? So what's going to happen then is you're going to attract histone acetylation binding proteins, and one of the most well-known of those, or some of the most well-known of those, are the bromodomain proteins, BRD2 and 4. And interestingly, a couple of years ago, Chris Sansam showed in a 2 hybrid assay using a replication initiation protein as a bait, treslin. So treslin is a protein that comes in along with a bunch of others, and its specific job is to come in and activate the helicase, okay? So this is a bona fide replication initiation protein. And he finds that the top hits in a yeast 2 hybrid for interacting with treslin are BRD2-4. BRD2-4 happen to bind to histone acetylation, and they also happen to phase separate. So BRD2-4, when it reaches high concentration, it's going to drop out of solution and form a liquid-liquid phase separation droplet, okay? And then, by this logic, it's going to attract a lot of treslin. So therefore, we have a microenvironment with a ton of an initiation protein in it, just sitting there in G1, waiting for S phase to be activated when the cyclin-dependent kinases come up. And so we propose these are going to be the first regions in the genome to initiate replication. Okay? So that's our current working model. All right, last story now. We're at 53 minutes. This should only take a couple of minutes to complete. What we haven't talked about is the biological significance of a replication timing program. So it doesn't make a lot of sense that you should have to duplicate the genome in a particular temporal order just to get the DNA replicated. Okay? So I've always thought since an undergrad, this is something that I've been working on since an undergrad, actually, I wrote a paper on. Actually, this model was in my undergrad paper. That it must be for something that transcends the basic need to replicate DNA. And since we know, again, de facto, by the way, I have a philosophy degree, so you will see me put logic down this way. We know de facto that chromatin is assembled at the replication fork. We can't deny that, right? And it's torn all the way down to the nucleotides and then reassembled all the way back up. So my feeling, so that's the facts. And then my instinct is, why would Mother Nature waste that opportunity? She's completely erased chromatin. Why would she waste it to not either maintain chromatin if you're the same kind of cell or change it if you need to change it, right? That would be the place to do it, right when you're building it up. And we know that different types of chromatin are assembled at different times during S phase. So the model was that early, different types of chromatin are assembled and that replication timing controls the epigenome. But I had no way to prove it, not as an undergrad, not as a grad student, not as a postdoc, and not for the first 20 years or more of my independent laboratory. Because, as you might recall, I told you that we insulted cells with everything we could, every kind of knockout, every kind of drug, and we couldn't perturb the program. We had no means to mess up the program, so we couldn't do a necessity experiment, knock out the replication timing program, and see what it's necessary for, right? Until we came across a protein called RIF1. And you can ask me about how we got to the RIF1, et cetera. But all I'm going to tell you today, and this was published last year as well, is that RIF1 coats the late replicating compartment of the genome. And when we knock it out, we get, in some cases, what looks like a complete disruption of replication timing. We get a delocalization of histone post-translational modifications, and we get a disruption of the 3D organization of chromatin. Okay? And this work was done by a couple, both in life and in the lab. Peiyao Zhao, who is a computer whiz, an amazing coder, but she's not very impatient at the lab bench. And Kyle, who he's the best experimentalist I've ever had, but he hates computers. So these guys are a perfect couple in every way. And I could tell you anecdotes about, but I don't want to just take too much time. So, okay, so I'm just going to show you two experiments from this paper. The first one is the initial observation. At the top is the high resolution Repli-seq that we talked about earlier. This is in human embryonic stem cell line H9. So that's a very typical Repli-seq profile. And the second track is what it looks like when you knock out RIF1. It's almost a complete snowstorm. You can't see any resemblance of the normal pattern. And so then what we did is we went on, and some of you guys know Victor Corses, because I heard he was a guest here once. So I had a grant with him for 5 years, a long-term collaborator. And so he had somebody who was really a magic chip person, chromatin immunoprecipitation person in his lab. And he's also a great guy to talk to about anything. So we collaborated, and we looked at, we did chip of a number of different histone post-translational modifications. Shown here is H3k27 acetylation. So you can see in wild-type cells, you see these large peaks of K27 acetylation. They align with the earliest replicating regions in the genome. When we knock out RIF1, we get a delocalization of these. We lose a lot of these peaks, and we get more acetylation in other places. We can show that quantitatively by spike in chip and by Western blot that we're not losing H3k27. It's getting mislocalized throughout the genome. And we showed that for several other histone post-translational modifications. And what I'm going to show you in the next slide is the data that shows, well, I'm actually not going to show this exact data, but we could show that this replication, by using a conditional knocked out of RIF1, we could show that the replication timing program was completely abrogated in the very, very first S phase after we lose the protein. So equally disrupted as a clone, a knockout clone that you've cloned up from a single cell and expanded, et cetera. So in the very first cell cycle, the replication timing program is disrupted. But the misincorporation of the post-translational modifications occurs gradually with each cell cycle getting worse and worse with time. So you know that it's now a kind of a random program. That means at random, some regions are going to replicate at the correct time. So you're not going to get a complete wipeout of the epigenome in the first cell cycle because only some regions are replicating at the wrong time. And then we see alterations in compartments, and we see gradual stochastic and cell type specific effects on transcription. So the reviewers are really giving us a hard time. How do you know RIF1 doesn't affect transcription, which then affects replication timing? Well, what we find is that in the first cell cycle, there's only a few genes that are affected. If you do single cell RNA-seq, they're different genes in different cells. So these are just cells that are getting disrupted from the replication timing changes at different locations, and they're having different genes that are being affected. Okay, so we think altogether we can make a case for causality for the first time ever, that replication timing has a function and that it functions to maintain the epigenome. So this just shows you that experiment I just described. We used HCT116 cells because they are easily synchronized, easily manipulated. And there is a guy, Masato Kinamaka in Japan, who has made conditional knockdowns of every interesting protein you could imagine, fusing them to the plant aid domain, which allows you to add auxin, a plant hormone, to mammalian cells, and the protein that's fused to that aid domain will become degraded. And in the newest version of the system, you can't detect any protein within 30 minutes. It's a very nice system for mammalian cells. 30 minutes is a short period of time. So then we basically synchronize the cells in G1 first, and then we knock down RIF1. And then we can either hold them in G1, don't let them go into S phase, don't let them start their replication program, or go ahead and let them in, okay? And then we'll let them go through S phase, and then we do the chip of the different post-translational modifications. So I'm showing two of those here, and what's in blue are the peaks that are going to be affected by RIF1, okay? And in black is the rest of the genome. And what you can see is that if we hold the cells in G1, don't let them go into S phase, we don't get any effect on the post-translational modifications. So they are not direct effects of losing RIF1. Okay? But you need to get into S phase and sort of into mid-S phase before you start to see the post-translational modifications change. And if you look from cell cycle to cell cycle, they get more and more intense. Okay? So now here's my last slide of the talk. Oh, I did use an hour, didn't I? Darn it. I can give this talk 3 times. It'll be a different amount of time each time, depending on, I'm reading you guys and looking if you're puzzled and explaining more if I see a puzzled face. So at any rate, I want to leave you with an interesting question. So if we can disrupt the epigenome and the cells are perfectly happy, they're growing, they're healthy, we can expand them, and pluripotent cells, human embryonic stem cells, retain their transcriptional regulatory network, they retain their pluripotency. We've shown that in very short experiments that they can go to endoderm, mesoderm, and ectoderm. Then what the heck is the epigenome for, right? So there are two observations here over there on the right. One is that we don't affect cell identity by knocking out RIF1, even though we affect the epigenome. Secondly, though, if we look at mice that are knocked out for RIF1, they will die at random points after gastrulation. And so that tells us that RIF1 is an essential gene for development, right? So the hypothesis that we're proposing now is that the epigenome is largely dispensable for self-renewal, but is required for lineage commitment. And we have over many years, I had a 10-year program project grant with where we developed methods to differentiate human embryonic stem cells into all sorts of different cell types. And we can see various intermediates going to pancreas and liver, et cetera. So what we would predict is that there's going to be a critical point where the replication timing program is going to be important. And using the systems that we have, we can knock RIF1 out, and then we can bring it back later. Or we can knock it down at different time points and find out where the critical period is. And we believe from results I didn't have time to tell you that there are other things in RIF1. So that there will be some cell types that will be resistant to the loss that will be independent of RIF1. So we hope to dissect these somehow in the future. But I'm going to need a pair of hands for this one, by the way. Okay, so this is my current size of my group. It's quite small because we just moved from Florida to San Diego last year. I still have 3 graduate students left in Florida, Jesse, Dan, and Ethan. And I'm sort of rebuilding the lab in San Diego. Ronnie was my first postdoc. Marie actually moved as an undergrad from Florida to San Diego. She's now one of my techs. And Mackenzie was another undergraduate. She just can't seem to leave the lab. She comes to every lab meeting. And Rob is a temporary tech. He's going to go off to medical school. And Takayo celebrated her 20th anniversary with me last year. So she, of course, moved with me. And she and I are kind of the core of the lab. So we're rebuilding. And I just wanted to show you our new building. I had to move twice because the building wasn't ready. So I moved in July, and then we had to move into our new building in September. So it's been kind of a lot of delays in that sense. But I want to point something out. I went to work for this place because it's small, and it has nothing else to do there but then science, okay? There's no committees. I mean, for you guys, students and postdocs, you don't get involved in that. But for people like me, there's always distractions in universities. And if you have students, you have student programs, et cetera, et cetera, et cetera. So I went here because I can do nothing but science now for the rest of my life. And the only thing I have to do is pay all my bills myself, no subsidizing from a university. So that's what we do. This was started by a woman, Joanna Davies, who was at Scripps, one of the bigger soft money institutes, run the same way, but they're so big they have committees and they have problems. So she left and branched off and said, I'm going to build something that's never going to get that big. So we're going to try to stay in the 12 PI range, plus or minus. And this is our new building overlooking, this is the opposite side of the ocean would be behind us. But I want to point out that she started this 7 years ago. And last year we were ranked number 7 out of 100 funded institutes in San Diego County. So that's pretty good CEO. So it's going to be a little hard to break 6, but I think we can get up one more a little higher. But we're expanding, we're hiring faculty and hiring postdocs, so if any graduate students are interested. And also those undergraduates looking for internships, that's something we can definitely do. What we don't want to do is start all of the administration of a graduate program. So that's the only thing we're not going to be doing. Okay, and that's everything. And I can take questions now. Do you want me to leave any of these slides up? Sorry, I didn't show a beach picture. There you go.
Well, first we have a question in chat. This is a 2-part question, and it's from Patricia Leon. It says, what happens to ERCs in cells like neurons that are not going to replicate the DNA? And the next part is, if you affect the ERCs, something happens to the chromatin organization in these cells?
Okay, so I mean, the second one is that definitely yes, because we disrupted the ERCEs. Well, what we showed was that we lost all transcription, we moved to the periphery, and we moved to late replication. But we didn't really look at chromatin. We would predict, so actually, we didn't do the exact experiment that you're asking for. Although we are doing this now because we now have a system where we can induce a shift of a whole domain from late to early or early back to late. And we're doing the chromatin organization of that. But I would bet anything, my career, that when we knock those ERCEs out, that we get chromatin changes. Your first question is about terminally differentiated cells that have withdrawn from the cell cycle. I would guess under those circumstances, they're not going to care about ERCEs replication function because they're not replicating. That would be my prediction. ERCEs seem to be clustered elements that have a lot of other things in them. So that's something we still have yet to decipher. So if you deleted the ERCEs as they are, as they were in the first paper, the only paper that's published, you might affect other activities such as transcription. But I would predict once we get down to the elements, I mean, Jesse has a couple of them down to 150 base pairs. Once you get down to that level, that they wouldn't matter, but you may need those cells sometime to re-enter the cell cycle and fix tissue damage or for regeneration or something like that. But once indeterminately differentiated cells, they should no longer need that information to replicate, for a replication program. Interesting questions. Yeah. Yeah.
So, can you hear me?
Leave it on.
So, yeah. So I was wondering about
I can hear you.
also about the RCs and the oct, hora, nano, and SOX. And do they recruit MCMs, or does MCMs recruit oct, nano, and SOX? I mean, because there seems to be like a correlation, a direct correlation between these factors and MCMs, right? In order to get replication going. So have you looked at any direct interactions between these proteins?
Yeah. So the question is, does the helicase itself recruit OCT4 SOX dynamic? Could be the other way around too, right? Yeah. Well, first of all, we don't think that replication is initiating at the ERCEs. We don't think the ERCs are origins, right? So again, the analogy, people are always want to think that, but the analogy is quite simple. Think of transcription. The enhancer's over there on the wall, the promoter's over there on that wall, right? But they're talking to each other and they're both important for transcription. We think we're looking at the enhancers transcription. So of replication, sorry. So they're regulating when replication is going to initiate, but then they're forming an environment within which where doesn't matter. Where you actually initiate doesn't matter. In transcription, you might imagine that the ERCE would be talking to the origin, and that's possible. That's still possible. Maybe interacting, although with transcription, enhancers and promoters don't always interact. So we think they're independent. But to get to your question, I think the first thing we have to do is be able to map MCM before we can answer any of those. Have people seen in pull downs, does MCM come down with ORC or come down with, MCM come down with OCT4 SOX nanog and vice versa? To my knowledge, no. There is occasional anecdotal papers showing MCM coming down with MIC. And it's really controversial because some people think it's a joke and other people see it, in different systems. But that's all we know. That's all I can tell you.
Okay.
And
so the other one is, these are the pluripotency factors, no, that you use to reprogram. So then turning late replication genes to early replication genes would be a requisite to reprogramming also.
Yeah, absolutely. Yeah. When you overexpress OCT4 SOX2 nanog, our model would predict that would drive switches to early replication. Yeah. And the partially induced pluripotent stem cells that I told you about fit with that model. Because for some reason, the Yamanaka factors did not push some regions back to early replication. And those cells can't reprogram. Yeah, that would all fit. Yeah. Other questions? The undergrads asked 100,000 questions yesterday.
Okay, next question in the chat comes from Arnaud Roncelet. He says, thanks for this very nice talk. Do the early replication sites map close to human hotspots of meiotic recombination?
Do the early replication, do ERCEs map close to
human
hotspots of recombination?
Yeah.
I don't know. Show, you can look it up. We can give you the list of ERCEs. I don't know. I've never looked at that. Yeah, I wonder if you'd like in the chat box to, I don't know where I should be talking to, but if you'd like, you can tell me what your hypothesis is, because I'd be very interested to hear it.
Oh, sorry. Is RIP1 a single active molecule, or does it have cofactors?
You like
order
RIF1.
Yeah, back to Rick.
I'm having a little hard time hearing you between the mask and the volume. Something's muffly. If somebody else can interpret.
Yeah, I'm very sorry about it. Again, with RIF1, is it a single acting molecule or does it have cofactors in order to unleash the effect of regulation?
So does it act on its own or does it have, okay, so RIF1 is a 270 kilodalton protein. It's huge. And it has a big N-terminal region that's full of heat domains. And these heat domains are known to be attracting all sorts of proteins. So I would suspect that RIF1 is a scaffolding protein that brings in all kinds of things. Now, RIF1 is also, it's most well known for its role in double-strand break repair, actually. Okay, so it prevents homologous recombination in G1 before you have sister homologs. So unless you have two copies right next to each other, right after replication, you don't want to do homologous recombination because you don't have a homolog. So you suppress homologous recombination in G1, and then RIF1 sort of stops doing that during S phase, okay? So that homologous recombination can occur. And that's where it's most well studied. So it has at least those two roles. And then there are proposed roles, other proposed roles that people have come up with. So that's how, I mean, so that's speculation that it's scaffolding a ton of proteins, and it's very insoluble. It's hard to chip. Now, what we know about how it makes regions replicate late, is that kind of part of your question?
Yeah.
What we know is from budding yeast and a little bit from mammalian cells, a paper that we co-authored about a year ago. So from budding yeast, it's been shown, 3 independent labs showed that RIF1 on its C-terminus has a PP1 interacting domain. So that's protein phosphatase 1, okay? And protein phosphatase 1 removes phosphates from the same motif that the initiation kinase, CDC7, the devoted, sometimes called DDK, it phosphorylates MCM, and that helps to activate the chelicase. So it's dephosphorylating the same motif that the activating kinase is phosphorylating. So RIF1 antagonizes initiation by way of bringing in a phosphatase, okay? That's been shown pretty tightly in budding yeast. And in mammalian cells, it also has a PP1 binding domain. That binding domain can be mutated in vivo, and those cells have a partially disrupted replication timing program. So it seems to be almost everything in budding yeast, but half or a little more than half of RIF1's activity on replication timing in mammalian cells. So what the rest is, we don't know. Does that give you enough? You didn't ask me how something that's localized to late replicating regions can affect early replicating regions. You didn't ask me that. So I'm not answering that yet.
Okay, so next question from chat comes from Luis Covarrubias. He says, it was not completely clear to me if RIF1 is important for chromatin architecture or to define the DNA replication pattern.
Okay, so what we know is that replication is disrupted before the three-dimensional organization of the chromatin, based on all the measurements that we've done so far, which is pretty deep high C, but all kind of genome-wide analyses. We haven't gone in, tried to carve out specific parts of the genome and see if there's effects. So while we don't know exactly why, I just answered partly why the replication timing program is disrupted immediately when RIF1 is gone, because now PP1 is no longer concentrated in late replicating regions. What we do know is that that's the first effect. That's the primary effect of knocking RIF1 out. So In fact, the really, I don't even think there's any detectable, it's a little, I'll tell you that, I'll just have to say it now because I've started. So wild type cells undergo 3D conformational changes of chromatin when they enter S phase. Those 3D conformational changes are abrogated by RIF1. So I was about to say RIF1 has no effect on 3D chromatin in the first cell cycle. What it actually does is prevents an effect that happens in wild type cells. Okay. So that's about as much as I can tell you as evidence that the 3D changes are downstream. They're happening in subsequent cell cycles. So the interpretation of that is first you got to mess up the epigenome, and it's actually the epigenome that's holding those other 3D things together. We do know that when we lose H3k27 peaks, when they get delocalized, that normally they interact and those interactions disappear. So that's again consistent with disrupting the ERC interactions by the RIF1 protein that's not anywhere near the ERCs. So, yeah.
And the next part of his question was altering cell cycle checkpoints, for example, by oncogenes or tumor suppressors, affect the DNA replication pattern?
Can you repeat the first part of the question so I make sure I got it? Yes.
Does altering cell cycle checkpoints affect the DNA replication pattern?
Okay. So I thought there was something maybe about oncogenes in there, but so the checkpoint. We showed actually by cytogenetics, the methods I talked about in the middle of the talk, that disrupting the checkpoint does not affect the temporal order of replication. What it affects is how many sites are firing synchronously. So you could have the replication timing parameter compressed by disrupting the checkpoint, but the temporal order is still preserved. And one of the ways we showed that was we inhibited DNA synthesis. First we labeled with one color analog, like a nucleotide analog. Then we blocked replication with a phytocolon very hard, so like really a hard block of replication. And then we either knocked the checkpoint down or not. We knocked the checkpoint down and let it go 1 hour, 2 hours, 3 hours, 4 hours without a checkpoint. Replication can't get going because we've got the inhibitor drug in there. But when we release the inhibitor drug at those different times, replication picks up at the foci that were scheduled to be replicated at the amount of time that the cells were in the checkpoint inhibitor. So it's a really remarkable paper. It was in Nature Cell Biology in the year 2000. And basically that shows that this program has a clock and that the checkpoint is just trying to keep the clock on target. But that without the checkpoint, you're still going to get assembly and disassembly of the proteins at the correct sites in the proper temporal order. So yeah, I think that answers the question. Yeah. Next. Yeah, we going like chat live. Okay. Mm-hmm. Yeah. So you had a similar question yesterday, and we might have even talked about this last night. I can't remember, but I think we might have. But so we don't have enough information to answer your question, right? So we can't really analyze. We can't do a motif search on a 40 kb segment of chromatin. So and our computational predictions were windows of 25 kb, okay, when we predicted 1,800 and something of these things. So one thing we're doing is getting them down to a small amount. And then we need enough of them so we can make a consensus. But we also need an assay that's high enough throughput for them to really get numbers. And then you want to mess the sequence up in lots of different ways, and you want to test them. So we need an assay, and as I used to tell undergrads when I was teaching, when you're doing science, everything is the assay. Your assay has to be robust. That means it has to be fast and interpretable. If it's fast but not interpretable, it's a waste of time. If it's interpretable and takes a year, it's a waste of time. It has to be a fast interpretable assay. So even with CRISPR, it's kind of slow. So we're trying to develop the insertion assay that I showed in one of the slides. And we're trying to get that to be as high throughput as possible. Then we can come back to your question, right? We can put 100 different elements in there and measure their activity. And then we can do sequences. If there's not a sequence consensus, we can look for a structure consensus or a melting consensus or any of these sort of different ways you can look at DNA, which I think is what you're getting at. Yeah. Give us a little more time. Yeah. Laura might be doing this. Because that's exactly what we want to do with Furhat. Laura's working with Furhat.
So a second question from Arnaud from Seret. How conserved are the DNA sites of early replication in other species, such as plants?
Well, conservation of plants is tough to do. I'm not a good enough evolutionary biologist to do that. So, but we have done a couple of related things. We had a paper some years ago comparing mouse and human, and we showed that the regions of the genome that are constitutive, in other words, if they're the same in every cell type, they're highly conserved across species. If they're developmentally regulated, they're much less conserved across species. Then we had another paper comparing a whole bunch of different primate species. I called it the Jungle Book paper, and actually wrote a song about it, which picked up by the radio. But anyway, kind of funny. But we compared orangutans, chimps, gorillas, I forget, it was 7 different primate species. And in that case, the analysis was done by a really high-end computational biology colleague of mine, Jen Ma, in Carnegie Mellon. And so the bottom line is it's highly conserved, but you can find differences, as you might expect. Now, going to plants, I'm not sure exactly what question is you're asking, but plants do have a replication timing program. It does seem that their euchromatin is replicated first and their heterochromatin last. They do have replication foci. The foci patterns, the spatial patterns, have been shown to be conserved from Hydra. You guys remember Hydra from, I don't know, maybe high school or something? This little organism that has just a few tissues. And even in Tetrahymena, which has, and so the way I think about it is Tetrahymena has a 100 megabase genome. So you don't see these things in yeast, but the whole genome of yeast is like one tad, of a human. So Tetrahymena have a 100 megabase genome, and replication takes place in the interior early and around the periphery late, and then Hydra, and then it goes up to, I forget. There's a review article that shows the stainings of early and late in all different species. So that part's conserved. In plants, plants have some big heterochromatic domains. People have looked at replication timing. Plants often go tetraploid for certain stages of their development, or endo reduplication. And I think that's been looked at. I'm saturating my knowledge here. I was on a NSF review panel for a plant group, and they were studying Arabidopsis replication timing to try to get, more genetic, kind of genetically manipulable information. And all the basic principles were the same. They kept, it was kind of, I don't know, it's, I would always ask, why are we doing this? It's all the same. And the answer would be because they're plants. So but plants look pretty similar. Hope I answered the question, because I don't know how you would look at really conservation like of the sequences.
Okay, next question comes from Mario Zurita. What happens? What
Ah,
happens if you
Ask him if his daughter graduated. Did she pass her PhD?
Oh, yeah, she did. Yeah.
Yeah, she got it?
That's why
Okay,
he's
good.
not with us.
Okay, so now we can relax.
He says, what happens if you incubate with JQ1 with the hub in ERCs?
Ah, okay. Wow. So this is a mystery to us right now. You know, you're opening up a big can of worms, all really new stuff. So not so new is the JQ1. We treated with JQ1 student Jesse. He got interested in this a couple of years ago. Different concentrations, different. Anyway, we could see no, significant effect on the replication timing program by treating with JQ1, but we could see BRD4 foci being disrupted and things. So we didn't know what to, it's a drug. We didn't really know what to think of that result. So we put it away. And then recently, Raj Jain and University of Pennsylvania made a degron of BRD4 in mouse embryonic stem cells, and we looked at that. And again, we do the degron of BRD4, 24 hours before the cells are looking like they're going to die. And there's a long time, plenty of time to change the timing program. And I mean, there's some, you can squint and you can see some effects on timing, but there's nothing global happening. There's nothing major happening anywhere. So that kind of, and that's a very recent, we're talking about 2 weeks ago that we got that result. So now we're starting to think, okay, we've done the drug and we've done the protein. Maybe the BRD4 thing is a problem. But just about a week ago, a paper came out in Nature showing that BRD2 knockdown wipes out compartments. And BRD2 and BRD4 compete in their activities with respect to genome architecture. And there's a study by Raj Jain showing that BRD4 is required to recruit NIPBL, which is what recruits cohesin. So that kind of puts BRD4 in the cohesin extrusion part of what genome architecture. And we know that compartments are the opposite, because we know when we knock out cohesin, compartments get stronger. So in this paper, they show that BRD2 knockdown wipes out compartments and it competes, it has opposite effects in many different things that they measured with BRD4 knockdown. So now we're going after BRD2 is the answer. So BRD2 was an equal partner coming out of the 2 hybrid with Treslin. So we just might have been, I think I say BRD4 more than BRD2 because it's more popular. It's known to be involved in many cancers. So it's a more popular molecule. But maybe BRD2 is our guy. So you opened up a little Pandora's box there, Mario. That's all confidential, right? So turn off the recording, But hopefully we'll have the answer soon.
Okay, next question comes from Arnaud. In yeast, Valerie Board showed a correlation between replication and meiotic hotspot of recombination. And I wonder if it can be observed comparing your rep-seq data and hotspot regions from humans.
Okay, so can you, I didn't quite hear a couple of the components there. So somebody showed in yeast that replication timing was related to, was it recombination?
Meiotic hotspot of recombination.
Meiotic hotspots. Oh, is this the recent Cell paper? Or is this in yeast? Did they say in yeast?
Yeah, this was in yeast. Yeah.
Yeah, because I don't know if the recent Cell paper shows that much, but they do look at replication timing and they do look at meiotic recombination in mice. So it's an interesting paper. If you haven't seen it, it's in Cell. I reviewed the paper actually. So I don't know this paper you're talking about. Okay, I would just say that when you see a correlation, you sort of have to be careful because things can be correlated for all sorts of reasons. They can be correlated causally, but indirectly separated by, 6 things that this is related to that, this is correlated to this, and this is actually quite common in genomics. So the meiotic hotspots could be correlated to replication timing because they are, associated with chromatin that's associated with early replication. I don't know what the exact observation is. They could be, in yeast, you can get a little more precise. They might be near certain arse elements or something. I don't know, but I don't know exactly what the result is. I'd be happy to talk about it, off later when, yeah. That's the disadvantage of Zoom.
Yeah.
Unless we can bring the person up on the screen.
And the final question comes from Irene. When you knock down RIF1, do you also alter other epigenetic marks besides acetylation, like methylations? And how long do these alterations persist? Can you revert to the correct epigenetic marks by providing RIF1 back?
Yeah. Okay. So first of all, we looked at 4 post-translational modifications of the paper. Pretty sure it was 4. So H3k27 acetylation, H3k27 trimethylation, H3k9 trimethylation, and H3k4 trimethylation. Okay, so we're looking at an enhancer mark, a facultative heterochromatin mark, a constitutive heterochromatin mark, and a promoter mark. Okay, that's what we're looking at. So the H3k27 data I showed you, the H3k27 acetylation data I showed you, the H3k27 methylation, trimethylation data I didn't show you, shows also alterations, but they were interestingly, we looked at 3 cell lines in that paper and they were different in the different cell lines, which might not be a surprise for a facultative heterochromatin mark, but they had different sort of effects. And then H3k9 trimethylation, there was something very interesting there, which is that, and you can read all of this in the paper, but there's very little broad H3k9 trimethylation domains in stem cells. There's very few. It's a tiny percentage of the genome. But in HCT116 cells, which are, adult, they happen to be colon cancer cells, but they have a lot of big H3k9 ME3 heterochromatin domains. So like lots of cells do. And what we found was that some of those domains were not affected for replication timing, so they stayed just as late. And what happened was they gained H3k9 trimethylation in those domains when we knocked out RIF1. And then there was another class of H3k9 trimethylation domains that were affected by RIF1 knockout and lost H3k9 trimethylation. Okay. And then the other mark, H3k4 trimethylation, I'm actually blanking on what we saw there. So that's a promoter. I know we saw some changes, but I don't remember what sort of pattern that they were. So I'm just going to end there because I'll be sitting here thinking to myself, trying to remember. Now, the other question is, what happens when you add back RIF1? We haven't done that, but we can do it. So whoever it is, if there's a graduate student, they should come to my lab as a postdoc and do that experiment. So what what Dan might do before he takes off on a job, he hasn't decided where he's going yet. He's getting all these job offers, but he might do that experiment. I just don't know if he's going to have time. Students think they're going to do everything, and then, once they get a job offer they like, that's where their head's gone. But he has fantasies of doing the experiment of knocking down RIF1 and then doing single cell TIP-seq on histone modifications. And in a single cell where we can see whether a region has replicated or not replicated, and then say it's replicated at the right time in this particular homolog. So we have to do it in the mouse hybrids so we can see homologs, because otherwise it's the average of 2 and we'll never see anything. So this homolog this region has replicated at the right time, and it re-put the correct histone post-translational modifications on. This other one in the same cell replicated at the wrong time, and it put the wrong post-translational modifications on, right? and then collect all that data for thousands of cells in, every 50 kb in the genome, and then bring RIF1 back and do it again. So I think that's this experiment that this person wants to do. So, but I don't think Dan will do it. So you're still welcome to come to the lab, and he certainly won't finish the story.
And
More? I'm going to need some mezcal here pretty soon.
Well, have you seen replication timing in heterochromatin? I mean, it's late replication, but have you found some early replicating elements in there, or some hubs that are different with that timing?
Yeah, you can find exceptions to pretty much every rule. But first of all, you have to define heterochromatin, right? So what is it you're going to use to define that? But you can see large swaths of late replicating stuff with little regions coming out that are early replicating. You can find exceptions, things that are in the B compartment that are early replicating. You can find, I mean, there's no 100% correlation of anything to anything. Okay. It's usually like 80% is, wow, that's huge, right? So that means 20% of it is exceptions. So you can, pretty much everything you can think of. Like I said yesterday in your class, we have genes that go from late to early when we differentiate stem cells. Go from early replicating to late replicating when we differentiate stem cells, and they go from off to on, right? So they're doing everything exactly the opposite. So you can, there are these things that, yes, you can find exceptions. And those could be interesting. I mean, there's tons of stuff to mine. When you go into the exceptions, you go in with the instinct that something has to be different, right? So it can't be all the same, and then this replication is different. There must be something about it that's different that affects the replication. So exceptions to the rules are really interesting to look at. But you generally want to look at the rules first to publish. If you publish a paper of only the exceptions, you might have some trouble.
So I think that was the last question. So muchas gracias por venir. Fue un placer haber estado, que hayas tenido aquí. Y pues al final del día ya todos nosotros trabajaremos en Biomed, yo creo.
Yeah. I didn't actually hear that, so
that
went over my head.
Maybe later we're all gonna go work at Biomed with you.
Oh, okay, that's great. Yeah.
Yeah.
I texted Farhad yesterday. I said, you guys have quite the Cuernavaca mafia, at La Jolla Institute. And I said, I'm going to try to get you guys to the other side of the highway, to the beach side of the highway. So, and he said, sure, go ahead, try it. We'll see what happens. Anyway, let's get you all just to San Diego because there's so much great stuff everywhere there. So,
yeah, thank you very much for this.
But you can go to our website. I think there should be 8 or 9 faculty on there doing interesting things. Are we done? Shut down the computer.
That will be it. Applause, please.